I am Gülce Özdemir.
I am Hakan Ardayaman.
I am Zeynep Çelebi.
And I'm Mevsim Güneş. We are the students of Okyanus Science High School. Today we will introduce you our project, Bacteria versus Bacteria, Modeling of Burn Wounds and Burn Infection.
It is known burn wounds and post-burn infections are strong factors that can even cause death in these patients. The leading cause of death reported in the post-burn patients is the formation of a serious source of infection in the burn wound. For this reason, although various studies have been conducted in the literature to prevent burn wounds and infections, intense efforts are continuing to develop these applications.
Wound healing is the body's natural physiological response to injuries. Rapid healing of damaged tissue is a complex and dynamic process. This process consists of 4 stages including hemostasis, inflammation, proliferation, and remodeling.
One of the most critical stages of the wound healing process is the infection of the wound site. Infection of the wound site may cause patients to experience life-threatening complications. In this context, wound infections are mostly caused by biofilm-forming bacteria such as Pseudomonas aeruginosa, Staphylococcus aureus, Escherichia coli. The bacteria that causes infection can be highly resistant to various antibiotics. Failure to obtain results from antibiotic treatments makes the treatment of the infection in the wound area difficult. For this reason, alternative treatments are needed. In our work, we created a model to contribute to the discovery of treatments that can be applied to burn wounds and burn infections. For this purpose, we implemented a new burn wound and burn infection model in our study. To our knowledge, in our study, for the first time in literature, Tenebria multi larvae were used as a model organism for burn and burn wound infection. After creating the organism, we aim to examine the therapeutic properties of plant extracts and Lactobacillus, one of the most well-known beneficial bacteria in our body. In this model are Gotukola, medicinal plant used in therapeutic-based studies, wound healing process, antimicrobial, anti-inflammatory properties.
We would like to explain in the detail the method we used in our study. First of all, we took the bacteria we used in our project from our bacterial stocks and added them as cultures into 5 ml algae. We incubated it in an oven at 37 degrees Celsius for 24 to 48 hours. We planted the bacteria we obtained from the same culture into sterile agar petri dishes to prepare solid culture stocks. We incubated it in an oven at 37 degrees Celsius for 24 to 48 hours. Following growth in liquid culture and colonization in petri dishes, we ensured that the propagated bacteria were stocked at 4 degrees Celsius until the use in our project. In order to create vivo burn wounds in tiny biomolecular larvae, the larvae were cleansed one by one in separate groups. For this purpose, 70% of ethyl alcohol was gently sprayed on the body of the larvae. The petri dishes were left open and ethyl alcohol was waited to evaporate. And at the meantime, we prevent the larvae from coming out of the petri dish. After adding Pseudomonas bacteria into sterile agar petri dish, we incubated it in an oven at 37 degrees Celsius for 1 hour. We added Lactobacillus bacteria to the zone we opened and measured the diameter of the zone formed after 24 hours. The average zone was 3 cm and 25 mm.
The approximately 1 to 2 millimeter square vinyl we prepared to create the burn model was heated by holding it on fire. After the heating process, the prepared larvae were taken as a group. We have the heated mechanism on the body parts for 4 seconds and applied this process to all groups. We took the Pseudomonas bacteria planted in the petri dish into a sterile laminar cabinet, and then we lit a fire to ensure sterilization for the bacteria in the cabin. We took the larvae groups with which we created the Pseudomonas infection into a fume hood cabinet and prepared them for infection. The larvae were removed one by one from the petri dish they were in, and we performed infection treatment in another petri dish. For this purpose, we selected a colony from the petri dish containing Pseudomonas bacteria and applied the bacteria to the area where the burn wound was created in the larvae.
After the burn was created, we added some of the rough living environment to the larvae to prevent them from being further stressed and took the larvae into the fume hood cabinet. Then, we placed the larvae in appropriate petri dishes to apply the Lactobacillus bulgaris following treatment. Afterwards, we selected the Lactobacillus bulgaris colony that we had grown in agar petri dish and applied it to the area where the burn model was created in Tenebrio motor larvae with a 200 UL stereo stick pipette tip. Lactobacillus B following treatment, larvae, another experimental group, were both infected with Lactobacillus bulgaris. We also applied good cola extract. In this experimental group, the larvae were taken into the fume hood cabin, and after the Lactobacillus bulgaris treatment, we took 10 UL of the prepared good cola extract and applied it drop by drop with a stereo pipette tip to the area where we created the burn wound. In the results of our study, we demonstrated that we prepared burn wound and burn wound infection modeling. To the best of our knowledge from the literature, the burn and infection model in Tenebrio motor larvae was created for the first time in our study. The reasons for choosing Tenebrio motor larvae for this study are to evaluate its long lifespan, high chances of survival in difficult conditions, ease of maintenance, and their usability as a model organism due to their low production and maintenance costs.
In our project, the burn model was successfully tested using Tenebria motor larvae. Considering the durability of the larvae, during the application of the burn model, it was evaluated that these larvae were quite durable during the burning and subsequent stages, and that the survival of the control groups during the observed period was considered to be a very important factor.
According to results we obtained from our project, the creation of post-burn infection model with Pseudomonas aeruginosa was successful. The success criteria were determined by the observing sites of infection, such as redness, swelling, enlargement of the wound area in Tenebrio motor larvae's burn part. As a result, an average of 10% viability was observed at the end of the 5th day in Tenebrio motor larvae infected with Pseudomonas aeruginosa. Additionally, while the mortality rate in the group where burn wound and burn infection model created and treated only with Lactobacillus bulgaris was around 40%. At the end of the 5th day, the survival rate was 70%, with a significant increase in the group where we treated with Lactobacillus bulgaris and Guticula has been detected.
And lastly, these are our recommendations about this project. To investigate whether there is a synergistic effect by applying antibiotics to which Pseudomonas A bacteria are resistant and Lactobacillus B treatment in combination. To examine the effects of combined therapy with silver sulfadiazine, the gold standard in burn treatments. Examination of the results in Galeria melanola, the most commonly used model organism in the burn models in the literature. Observation of effects using different strains of Lactobacillus bacteria.
Thank you
Thank you for listening to us.